Okay, let's continue on with lecture 4.3b, where we continue on with our discussion of fungi, the medically important fungi. Now, when it comes down to it, fungi, the way we differentiate, the way we study them mostly is we look at their reproductive mechanisms. Later on in the semester, we will discuss, the actual infections. But for a microbiologist, a lab rat who's trying to identify a scraping or a sample that somebody's given me saying, here, identify this, we think this may be causing a patient infection. Well, we're going to look at how the fungi reproduce. It turns out they make different structures. Different species or different groups of species make different structures. So that's what we're going to look at. We're going to look at how they reproduce, and it turns out they reproduce via spores. Now, as you notice, above my head, is the statement. Fungal spores are not the same as bacterial endospores. Let me say that again. Fungal spores are not the same as bacterial endospores. Endospores are made within the bacterial cell, and it is foolproof. It is the panic room. It is how the bacterial cell can make sure the next generation, its daughter cells, are completely protected. Fungal spores? Not so much. Turns out fungal spores are going to be made in the thousands, the hundreds of thousands, the millions, and are just spread out with the wind, out with the drop of water, whatever. They're not made within the cell. They're made outside of the cell. And we're going to look at how that's done. Fungal spores are going to be either sexual or asexual. Here, when we talk sexual sex, we're not talking, the physical act of reproduction. We're talking about an exchange of genetic material. A, B come together, mix genetic material, and give offspring C. Same species, but genetically unique. Has all the same genes, but slight variations of. And we're going to see that fungi also reproduce asexually. So that they go through, they make spores that are genetic copies of the original cell. So what happens, whether it's a sexual or an asexual spore, they get released. And then that spore is going to land somewhere where there's water, there's nutrients, it's protected. It's going to germinate. It's going to open up. Out is going to come new hyphae. New hyphae is going to sprout out. It's going to continue to grow, branch, and given enough time, it's going to form a new mycelium, a new blue spot on the piece of bread, a new black growth on the surface of, a fruit. When we're talking the asexual reproduction, when we talk about reproductive strategies, and we're talking spore formation, let's talk the asexual spores first. These are going to be genetic copies of the original cell. We're going to see that there's going to be a spore-making structure. There are multiple types of spore-making structures out there. We're going to focus on just two for today, the sporangium and the conidiophore. The sporangium makes sporangiospores. Conidiophore makes conidiospores. Now, when you look at the sporangiospore, you're going to see that it's a covered structure. A thin, dry membrane covers all the spores, keeps them together. So that way, when a gust of wind, a drop of water, raindrop, or an insect goes by, it'll burst and all the spores will be released at once. This is different than the conidiospore. The hyphae, that little projection, is going to start dividing. It's going to make, long chains of spores. Kind of, when you look at it under a microscope, the kinetiospores kind of looks like a paintbrush, the end of a paintbrush, these long strands of spores. They can break off one at a time, release, whole bunches at a time. But it's released over time, not all at once, like the sporangiospore. When you look at the two pictures, up at the top, that's the sporangiospore. It's a covered, protective structure. Releases all the spores at once when they're ready. Whereas at the bottom there, you look at some different types of kinetiospores. It could be little round spores like the one in the middle, or it could be just, the hyphae just break off at the septate. Or it can, they can be in small packets, but it's still the hyphae separating. However it works. Turns out each individual species of fungi is going to have its own mechanism, its own method of doing this. So you look under a microscope, and you can look at and see, oh, hey, it makes sporangiospores. Uses a sporangium. This is only a certain list of fungi can do this. Or maybe it's one of the kinetiospores. Only certain ones can do this one versus that one. That's how you go and you can identify. Here you look on this slide, instead of an artist rendition, here are microscopic pictures. You can see that it actually does form a protective coating around them. All the spores are there until it ruptures and they're released all at once. Above my head, kinetiospores. Kind of paintbrush looking-ish. They can break off individually. They can break off as long strands, released haphazardly. We also can look at the sexual spores. The way that the fungi exchange genetic information is hyphae from mycelium A, hyphae from mycelium B. Has to be two separate mycelium. Will grow and they will come into contact. You see the picture above my head. A and B come together. They make this new structure. It's called the zygosporangium. A and B come together, make C. Same genes, just different versions of them. Genetically unique. They'll make hundreds, if not hundreds of copies within that zygosporangium that will break. Those spores will be released.